Greetings and welcome to the Dyadic International Conference Call webinar. It is now my pleasure to introduce your host, Mark Amelfarb. Thank you, Mr. Amelfarb. You may begin.
Great, thanks. Well, good afternoon, everybody, and thank you for joining the investor presentation. If you see the second slide, Safe Harbor Statement, Forward-Looking Statement, and then on slide number 3, we start getting into the thesis of the investment in Dyadic. So I want to make it very clear that the technology platform, C1, that we've used for 2.5 decades to make industrial products is now being refined and leveraged into the animal and human health. And the opportunities that we were pursuing before the coronavirus outbreak showed up are all ongoing and continuing. And there's a lot going on animal health. I think you know that we have 3 out of the top 4 animal health companies funding research projects on different vaccines and proteins. We're working with more than 5 of the top 25 pharmaceutical companies on developing technology for different proteins of different types and classes. We finished the Sanofi project, and it's under evaluation, and those conversations are ongoing and continuing to see what may be the next steps there. We're working with the Serum Institute of India on a variety of different antibodies and different proteins for different disease states, and those are ongoing as well. And then we are working with, you'll see, IIBR, the European scientists, and others as we get into it on coronavirus activities on what we think we have the gene candidates that are as good or better than the big pharma companies' candidates that we believe we can produce quicker, faster, and cheaper at extremely large scale. Let me go to the next slide. Slide number 4. We're very well financed. I think, in fact, there was a publication that came out about a week or 2 ago that talked about Dyadic being one of the 100 or so companies on the biotech that is actually well financed to survive and last and prosper and potentially grow through the episode we're all dealing with here and the financial crisis incurring through the coronavirus outbreak. The market capitalization, the stock price, insider ownership. I want to remind you that we have a very large insider ownership, and where me and my family own a significant portion of that, and we are shareholder friendly, and we're not looking to raise capital and dilute our shareholders. We're looking for partners to invest non-dilutive capital into the projects we're working on as we continue to advance the technology, the science, and the products that we're working on. On slide number 6, we've talked about this. We're mainly a European-based company from the R&D perspective, and the reason for that is the technology was incubated in Europe for the industrial side. The people that know how to program it, how to grow it, how to scale it to industrial scales, whether it be for industrial uses or biopharmaceuticals, are mainly European. Slide number 7, we have our management team. I think many of you might have met Ping Rossin, Matt Jones, Dr. Charlotte, and myself. We run very lean, and we do that intentionally because we have enough eyeballs on the technology between big pharma and animal and human health with governmental agencies all over the world. We're operating, obviously, now. You see, we recently announced a non-exclusive research license with WuXi Pharmaceuticals, Biologics. So we're in China, or we'll be in India, we're in Israel, we're in Europe, we're in America. We're working with virtually all the major players on the planet to try to really bring a new technology platform to bear on lowering the cost, speeding the development of biologic vaccines and drugs. Our board consists of some very strong supervision and corporate governance oversight. As we've talked in the past, Dr. Bose and Dr. Buckland are pharmaceutical executives that were in biomanufacturing of vaccines and antibodies for nearly 3 decades plus with each of them. And they have very vast contacts and relationships, and they supervise and oversee on an ongoing basis our research and development that's going on at VTT in Finland and BDI in Spain. And then Michael Tarnock, the chairman of the board, oversees the corporate governance along with the audit chair and Dr. Herbst. On the next slide here, corporate achievements and milestones for 2019. I'm not going to go over these in detail, but there's a lot going on. There's a lot of shots on goal. We have a lot of opportunity, and those opportunities are growing. And those opportunities aren't just growing in the coronavirus opportunity. They're growing in animal health. They're growing in human health. They're growing in a variety of opportunities. They're growing in our own pipeline. So there's development product pipelines going on in the metabolite space, in advancing the technology and the science. We've talked about glycoengineering and the success we've had in glycoengineering G0 and G2, human-like glycan structures. So there's a lot going on in a lot of different directions that can help us create enormous stockholder value over time. On slide number 10, achievements in 2020, we have, I'll get to the COVID initiatives a little later on, but you can read those there. There's multiple shots on goal there. We have 2 already going on, and we think we can have up to maybe 4 or potentially even more as people start evaluating the potential of the high productivity of C1 to make vaccines and potentially antibodies quicker, faster, and cheaper. And if they have the recipe with the gene sequence, we can help them put those through fermentation, expression, and we believe higher levels of lower cost to satisfy the global demand and the needs that people are going to have for that. In addition, we talked about WuXi. That's a non-exclusive research license. That means they're going to evaluate the technology. If you go back and look at their CEO's quote, he thinks it's the right time at the right place for WuXi. The world's looking for microbial fermentation. That part of the biologics business is growing. They're building a several $100 million microbial fermentation facility, and they're looking for new, better ways to put technology into that plant. And I think they believe, as we do, that C1 can solve and speed up the development and lower the cost of some of their funded projects with some of their existing partners and future partners. Animal health, we've been picking up speed there with, of course, 3 of the top 4 animal health companies we're already working with, and obviously we're talking to others. So stay tuned on that. And then the scientific achievements on the glycoengineering, we've knocked out 14 different protease genes. Instead of the cell growing slower, we're actually seeing it growing more rapidly, faster, doubling time. We have about a 2-hour doubling time versus about a 24-hour doubling time for CHO. So that's a big advantage. And we've also eliminated a lot of the protease background by about 50-fold from the beginning strain to where we are today. And that has to do with higher productivity and stability. So that's been a big, big step forward in both glycoengineering. So we're delivering on what we said we were going to do from a scientific perspective. That's going to open up more opportunities. The markets on slides 10, I'm not going to go through this. They're huge. If we can just succeed on 1 or 2 of the different projects we have, it'll be a huge success and lead into, I believe, opening up more doors with more companies overall across the spectrum here. C1 gene expression platform, as we've talked about in the past, this is a very, very robust, a very versatile platform. It's programmable, it's scalable. This is the reason why when we have entered into this pandemic that people are talking to us, because we can really change the game in biologics. We have the potential to do things that others can't. That means we can potentially make billions of doses of a potential vaccine for the coronavirus if we have the right recipe of the gene sequence. And we think in some cases with the scientists we're working with from Europe and with Scripps that we do have the right genes, and we're loading those genes and beginning to do that as we speak. Some are already loaded in C1 and some sequences are on their way. So the Israeli government we're doing that with already. We're loading their genes, the European gene, and we're waiting for the Scripps genes to load those. And we're also working with others, which we'll announce at the right time at the right place. So C1 really does have science, the leverage of the industrial power, and we're bringing that to bear not only in the coronavirus, but also on the traditional prescription drugs, whether those be vaccines or antibodies. So as you know, we obviously have succeeded, raised and generated non-dilutive over $130 million in capital on the industrial side, which culminated on 12/31/2015 with the $75 million we got in from the DuPont transaction, and subsequently bought $22 million of our own stock back. On slide number 14, you can see here the science. It really is all about this cell line, the serendipity, the mutation that led to several hundredfold higher productivity, the higher purity, the robust versatility in pH and temperature. This is the ability to program and grow and scale this cell is what's driving all of this. As we modify the cell to fit square peg in a square hole, and that's what we're doing with the glycoengineering. That's what we're doing with knocking out the proteases. That's what we're doing in staring at the metabolites. We're trying to modify the cell so that it meets the needs of our customers. And each one of those programs specifically is designed to do that. And in slide 15, we have all the genetic tools. We don't need CRISPR. We've developed our own versions of putting genes in, taking genes out, scaling this technology efficiently, effectively, getting quicker, faster, and better all the time. And when you compare that to CHO, once we've taken a gene sequence and we've loaded it into the cell, and we've now made a stable cell line, every time you want to run a batch, you can take it out of the freezer to the end of the batch. It's only 12 to 14 days. When you compare that against CHO cells, it takes them in the neighborhood of 41 to 54 days. So not only can we produce higher amounts, we can do it in 1/3 the time with much lower cost media. So it's win-win-win. As long as the gene's compatible with C1 and the purity and the quality and the performance meets the goals and objectives, and that's what we have to refine on different projects and programs. And in the vaccine space, it's more refined than it is in the antibody space, but we're working on both diligently and quickly. So we have the ability to do things, I believe, that others can't. And that comes to bear, which you'll hear later when we talk about the coronavirus. We ran stainless steel fermenters for 2.5 decades. We wanted to find out, can we also run single-use bioreactors? And the answer was yes. We virtually got the same productivity in a single-use bioreactor that we did in stainless steel. And if you recall, we've grown it up to 500,000 liter scale. I want you to keep that in mind because we're talking about here huge abilities to produce massive doses of vaccines at much, much smaller scale. So scaling is not going to be a problem with C1. We're scaling down. So how do we apply this to therapeutic proteins? We're applying this in the antibody space. We're applying it in the vaccine space, the virus-like particle space, the bispecifics or trispecifics. If you look at the trispecific number, the last time we showed this to you, it was 1.02 grams per liter. It was a bispecific. Trispecific, which theoretically should be more difficult, we now have demonstrated we've produced one at 6.12 grams per liter or 1 gram per liter per day versus the 1.02 grams per liter in 7 days in the past. So this thing's getting better all the time. The 24.5 grams per liter for MABs, the 1,780 milligrams per liter versus 30 to 50 milligrams per liter, which is what baculovirus produced in the ZAPI program. Baculovirus is what Sanofi's using in their coronavirus program. So we believe we can make 35 times more of it quicker and cheaper, potentially with better performance. If you look at the next slide, this is just showing you the binding of the monoclonal antibodies, IgG1s, IgG4s, was virtually identical when it came out of CHO and it came out of C1, and we have even more evidence of that in other programs. In the case of the bispecific, in this particular next slide, this showed you that it basically killed the technology neutralizing activity virtually the same. And again, we've seen more and more evidence of this, and even including in G0 and in G2 strains. And then, of course, we've showed this slide before, and this is for killing cancer cells. And virtually, whether you make it in CHO or C1, it killed the cancer cells in the same manner, in the same effective. So this slide is just kind of a graphic slide of the different glycoforms that hopefully will come up here, different glycoforms of C1. We started out with the one in the middle. That one in the middle actually seems to enhance potency for vaccines, so it may be better than G0 or G02. But we have all those strains today. We have the one in the middle that seems to be not only more powerful, more productive, but also potentially creates better immunogenicity to create more antibodies to neutralize the virus, and that's a big benefit. And then we can also put it into the MAN3, G0, and G2 strains, and we're now working on G0F and G2F. So the next slide, basically, we're talking about collaborations and how we're applying this technology. And because of time, I'm going to go through this very quickly. But we have different programs business objectives and opportunities, whether it's big pharma, where we expect to get cash up front, milestones and royalties with eventual licenses, like we did in the industrial sector with BASF, Abengoa, Shell, where we got over $30 million of upfront cash payments. And then we had milestones and royalties behind on products. And of course, DuPont then paid us $75 million. But also we're taking equity positions, and some of these equity positions could turn into very nice returns over time. And we don't spend a lot of time and effort on these things, but we hope that one or more of them, and we can do additional ones, but time is limited and our focus is more today on our own product pipelines, on the big pharma opportunities, and of course, accelerating the development of coronavirus vaccines and antibodies with the IIBR, with the Europeans and otherwise, which we'll get into. So the next slide is the ZAPI program, which we've talked about. So this is an important slide because this leads into why we're involved in vaccines and why the evidence, on top of the evidence we saw of the Sanofi Pasteur influenza program in the past. When we started this program, and this is the Zoonotic Anticipation Preparedness Initiative, this is exactly from animals to humans, which is exactly the problem the world is dealing with in this pandemic. But in this particular case, and we're going to compare it against baculovirus, and we have two data points, not just one. In this program, we made 35 times more of their antigen than they did in baculovirus. They've now done the animal study. Not only did we produce more, we produced it quicker, and it performed 100% challenge test. It was the immunogenicity was better than expected. With one single shot, we were able to immunize the cattle and the mice. And think about this: the dosage we used in zapping cattle, the amount we used, we'll talk on the next chart. Cattle are half a ton. That's a lot bigger than human beings. So we expect in coronavirus and other vaccines that you're going to use lower dosages. But as you'll see on slide 28, if we just take the dosage we used in the cattle trials of a half a ton animal versus a human being, big difference in weight, we can make 10 million doses in a 200-liter fermenter in 121 hours, or less than a week. Now, that's not making the final product because it's got to get downstream purification, and then obviously you got to put it into a way to deliver it. But in the fermentation part of what we play in and the role, we can help make, in this case, in a 200-liter fermenter, which is, by the way, the size of a 55-gallon drum, which you can do virtually anywhere on the planet, in any country, 10 million doses. If you want to make 100 million doses in a week, you can then use a 2,000-liter fermenter. If you want to make 1 billion doses in a week, you can go to 20,000-liter fermenter. And remember, we ran at 150,000 to 500,000 liters successfully before with C1. So whether it's 20,000 liters, 2,000 liters, or 200 liters, we can scale to those very easily and consistently based on the past history. So we can make 8 billion doses of the world's needs, depending on where you make them and what size, flexible commercial scale, within a month through the fermentation. So now let's talk about the collaboration with Sanofi. It's done in terms of our part. They've done analytics. They've looked at quality. They look at performance. They look at productivity. They look at stability. And so you got to remember here, these are things that are moving targets. When we started that a year ago and the technology we had then versus what we have today, it would be even better results today than we had then. And we met or exceeded more than half of the productivity levels that Sanofi asked us to at the beginning. And now those would be even higher. And I think you can guess that some of those numbers might be on those earlier slides. And they're evaluating right now, as I mentioned earlier, what the next steps will be with us and our scientists in the business development. Serum we talked about before, so I'm going to skip through this. The world's largest producer of vaccines by volume. We're working with Serum right now on some antibodies and other proteins, and those are going well, and that's ongoing and that's moving forward. The IIBR, this starts getting into what we've done in the past with governmental agencies, and we're speaking to U.S. governmental agencies as we speak, including even today, earlier, and ongoing about where they can help, what we can do to help them advance the coronavirus opportunity. The Israelis are involved with us in this for one reason and one reason only, 2 years ago, we started working with them to work on pandemics and epidemics and be better prepared for the problem and the pandemic we're facing today with coronavirus. The success we had with the Europeans in the IIB, or the Israelis in the enzymes, is the reason that they're now using C1 and testing it to be one of the potential cell lines that they may actually take in demand, which is what we'd like to do. And since time, I'm going to have to rush through this because I think we're running out of time here. We're working with not only the Israelis, we're working with the European scientists, the CR2O or Erasmus, Utrecht, TO Hanover. Those are 3 of the top 20 coronavirus scientific experts in the world. You can see from the footnote below. We're working with other programs. Stay tuned. We've talked about Scripps and the UFOVAX, and you can pick that up at another time. These slides here show you not only that we have the target, we're actually working towards producing those to get into animal studies, to get into ultimately toxicology, animal studies, phase 1, potentially phase 2. Same thing within the Scripps program. That's not as far along because they came in a little later. But again, it's not just about our targets that we have with Scripps, UFOVAX, the ZAPI scientists from the ZAPI program. We're offering this technology to anyone and everyone that needs help, that wants to mass produce billions of doses at affordable prices of vaccines, and we're working on that. And our technology, the different glycoforms, as we spoke before here on slide 35, are the reasons, not in addition to productivity and stability and high yields and low cost, is we can potentially put them in different various glycoforms to enhance potential performance, to make them better and more potent, so you can use lower doses. Slide number 36, you've seen before. We succeeded with Sanofi in the mice trials back in 2015 for influenza. The reason this didn't go forward had nothing to do with the science. The science actually exceeded expectations. And so this is data point 2. If you look at the middle of the 10 units per gram, we had 100% protection versus 50%. And guess what they used? Baculovirus. So whether they use C1 or not, it doesn't mean C1's not better, quicker, faster, cheaper, with potentially better potency. It could be political reasons or other reasons or comfort levels. But we're moving forward on this vaccine in these different programs, and we think we have a better route forward, a better way to produce larger doses at lower cost with potentially better candidates that may have enhanced potency by the benefits like you saw here in C1 in that mice trial. So basically, seasonal flu, we picked up the program with Oslo University, the world experts involved in HA, hemagglutinin, and other proteins for flu. So I'm going to breeze through that. I'll give you more of that later as we progress. So in the Oslo, we just talked about as well. And I'm just going to breeze through this. Certolizumab, we're working on figuring out what direction to go for this particular biosimilar, biobetter. That's the roadmap. Same thing for Opdivo, which we've talked about. That's our model organism for MABs as we do the glycoengineering and going from M3 to G0 to G2, and then ultimately G2F and G0F. That's the MAB in our model MAB, and that's going well. Metabolites is something that we're moving forward on. The secondary metabolite program is advancing. And then AAV viral vectors, that we've done put our toes in the water, but because we have so many other things going on, we're evaluating if and how to move forward there and with whom. 43 with the metabolites. And I think that's the summary, end of the presentation. And quickly, there's some Q&A, but I'm not quite sure how much time we have left. But it says, can you update us on the C1 fight against COVID-19 and about the announcement with WuXi? So basically, I think I talked to you, is we have multiple shots already on goal with the coronavirus vaccine opportunity, where we're loading in, we believe, some of the best potential performing vaccine candidates, the gene sequences, the recipes, into C1, which is the living cell to mass produce those. And we believe that we can potentially produce equal or better performing vaccines at a much lower cost at a shorter period of time once it's approved through the regulatory agencies. Next question I was asked, let's see here. The WuXi announcement, So WuXi, as I think you know, is a top 10 biomanufacturing company. If you don't know who WuXi Biologics is, you should look them up. They compete with Lonza, they compete with Catalent, they compete with Patheon. They work with numerous top 10, top 25 pharmaceutical companies, global. They're in China, they're in the US, they're in Ireland, they're in Singapore, so they're in Germany. So they're looking at C1 and evaluating it to potentially take a license, some type of collaboration, that's like venture. So once we put the system in their hands and they get to play with it and feel it and test drive the car, we hope they're like the Israelis and the Europeans, they'll feel the power of it and they'll want to get their hands on it and work together with us to create significant value for our shareholders. I think I'm being told basically I'm out of time, so.
Okay. Thank you, Mr. Emilfarb. And this concludes today's webcast. You may disconnect at this time. Thank you for your participation.